西部时间呢，西部时间是7点，中国时间是北京时间上午10点，就是1个半小时讲呢1个小时左右，半小时呢提问讨论。网站上就我们的网站上呢是会提前1天，有的时候提前几天发个信息，但是呢，提前1天呢一定是会把这个消息呢speaker 的名字啊，他们的内容的title啊，基本的内容介绍，这个会介绍一下。那么你呢这个只要注册1次，然后系统呢会自动发送连接，Zoom 的连接到你的呢email 信箱。但是我们非常鼓励大家能够你注册1次以后吧，它自动就不要你要去找，对吧？这个很方便，好吧？那么事情就是这样。现在嘛，请葛守仁博士呢来介绍呢，今天晚上有2个 speaker 他介绍一下。
Okay. Thanks, Professor Xun, to give this introduction. So today we are so happy to have 2 topics. Actually, you know, that's interesting because I didn't make this time quite good manager. So we have 2 speakers today. So first one is Yongjian actually from Texas A&M University. So recently, Yongjian actually published several very interesting papers using deep learning on the single cell data, especially for the genotype-phenotype association study. I think this is really important because we are going to have more and more single cell data, not only from RNA-seq, but also we have the AT-seq, you know, chromosome modification, and also genetic data from there. So identify the association between, you know, phenotype and genotype will be very important to identify, you know, always drug target, always understand the, you know, the disease mechanism better. So we are so happy to have, you know, Yongjian to give some presentation on his works. And for second one, we have Dr. Zhu. Dr. Zhu actually published a very good paper, paper called use causal inference to identify some causal inference from the single cell data. So this is a well developed software to identify this kind of causal relationship. So Dr. Zhu will use Chinese, but Yongjian we use English. So I noticed we have some audience is, you know, maybe you only can understand English, then maybe for the second one you can just leave. So yeah, okay, thank you so much. I will leave the time to Yongjian. So please start your presentation.
All right. So can everybody hear me?
Yeah.
So first of all, thanks for the invitation. And my name is Yongjian. I'm currently a 4th-year PhD student of electrical and computer engineering at Texas A&M University. My research is mainly about computational biology with machine learning. So basically, we are just developing and applying machine learning models on biomedical data, especially single-cell data, to try to provide the biology interpretation of translation between genotype and phenotype. Specifically, as my slides show here, my research focuses on 3 factors, which are cell-to-cell communication, gene perturbation, and gene activation, respectively, which are the key drivers for shaping the phenotype of an individual cell. They are affecting the phenotype leading genes, which will later shape the phenotype of individual cells. So today I would like to give you a brief introduction of the first 2 representative work of my research, which are cell-to-cell communication and gene perturbation. So in my first project, we developed a tool called the scTenifoldXct, which was recently published in Cell System in February. So first of all, I will give you a brief introduction of cell-to-cell interactions. So there are actually basically 4 categories of cell-to-cell interactions, which are autocrine, the cell regulating itself, and paracrine, the cell talk to its neighbor cells, and juxtacrine, meaning the close contact form of cell-to-cell communication. And finally, the endocrine, the long distance talk between two cells, usually through the vessel of blood. So why the topic of cell-to-cell communication matters? It turns out it can be applied in the field like cell, help people to understand the cell development, also the tissue homeostasis, and also the immune interaction in disease. Traditional way, people just use the fluorescent imaging to identify the protein-protein interactions. Although this direct measurement of protein abundance is preferable, but we know it's it's well known that the RNA-seq data is more numerous, and they are more easier to access, and they are straightforward to analyze, since we have already developed well-developed tools to analyze such data. In particular, the coordinated gene expression of a literature-created list of ligand receptors can be used as the proxy to infer the intercellular communications. And here we're gonna use the power of a single cell, the unpresented, the heterogeneity, to see what we can get from this single cell RNA-seq data. So here, these slides, I show the typical flow that people in the field of computational biology to use to infer the cell-to-cell communication from gene expression data. As the first step, experts or biologists will perform the single cell RNA-seq data, and we will use mapping algorithms to end up with gene expression matrix, which is a 2D matrix, gene by cell, that I think most of us are familiar with. And then we will query the database, the literature created database of ligand receptor pairs to subset this derived gene expression matrix. And then we will end up with a subset matrix, which consists of owning ligand receptor owning by cells. And then intuitively, most straightforward way is that we have to build a scoring function, which is a function for expression level of ligand receptor to indicate the strength of communication. So it turns out that people usually conventionally use the product of a ligand receptor expression level to indicate the power of communication. It's quite intuitive because the product of ligand receptor is actually a monotonic function. It increases as ligand receptor level increase. And then some of the tools also design the statistical inference to indicate the significance level of their inferred ligand receptor pairs. Since few study figure out what actually the product of ligand receptor located in what kind of a distribution. So people usually use here the permutation test to the non-parametric test to get the significance of ligand receptor pairs. So before I dive into my developer pipeline, I would like to introduce the two things that inspire me to think about my solution for the ligand receptor inference. First thing came from my senior in the lab. So my senior developed a tool called scTenifoldXct, which is a framework to construct a gene regulatory network by PC regression, principal component regression. So in this work, it utilized the manifold alignment to simultaneously project the two pairs of genes into a unified latent space and calculate the similarity of genes. But the thing is that the most important thing is that the PC regression by principal component analysis, we can significantly overcome the great sparsity of intrinsic single cell RNA-seq data. As we know, there are a lot of zeros in the raw count matrix. Another thing we know is that graphs, also known as networks, are actually matrices. So we know that a single graph can be represented by a matrix, but how we represent this the graph on my left-hand side with where there are two strongly connected components and a single correspondence, a single link between them. It turns out we can use the right-hand side matrices to represent the graph on the left with two highly connected graph sitting in the diagonal, while their correspondence is sitting in the off diagonal. So we see one here. This one actually indicates the link from node 3 to node 4. So with these two things, I developed a pipeline here. So the high-level interpretation for the pipeline is that we wanted to use the gene regulatory network level information to infer the ligand-receptor mediated cell communication. So as I mentioned in the last slide, we use PC regression developed by our lab to infer the single-cell gene regulatory network from two distinct cell groups we denoted by A and B. So we will have WA and WB, which are two distinct single-cell regulatory networks from two cell groups. And the big picture behind it is that we want to project the node from the network. Actually, in this context, there are genes from two distinct cell groups into a unified low-dimensional space. So here, the purple and the green dots represent two sets of genes from two distinct cell groups. And since they're in the low-dimensional embedding, we can calculate their similarity by simply just calculating the Euclidean distance between each pair of them. After that, we will end up with a distribution of distance. And it turns out that the highly communicated ligand-receptor pairs located in the left tail of this distribution. So we built our non-parametric test to infer the significant ligand-receptor pairs. And we also query the ligand-receptor database to validate our findings. So mathematically, the most novelty in this pipeline is that we propose to use two neural networks to solve the manifold alignment optimization problem. With some constraints. So as you see that we first constructed a joint similarity matrix here with WA and WB sitting in the diagonal and their correspondence sitting in the off diagonal. And we built a network and we allow the network to learn the similarity between provided by these joint similarity matrices. So more formally, we construct a W, which is a joint similarity matrix, just as I mentioned in the last slide. And the loss function in this manifold learning can be represented like this in this equation. It consists of basically, so briefly speaking, it consists of three terms. The first two terms indicate the inner similarity within two distinct cell groups. So this is the similarity within the cell group A, and this is the similarity within the cell group B. While the third term actually asks our neural network to also maintain the inter-similarity between cell groups A and B. So this is the inter-similarity provided by the correspondence matrix sitting in the off diagonal of the joint similarity matrix. So we also put some constraint to try to avoid the trivial solution of this optimization problem, and the solution edge can be represented as a vertical stack matrix with HA and HB sitting vertically with parameter theta A and theta B, which are the parameters of two neural networks. So that's actually the method for the single sample case. But actually, in the field of biology, people want to compare two different conditions and obtain the differential ligand and receptor pairs. Like we compare two groups of gene expression to infer the differential gene expression. So how to compare, how to obtain these differential ligand or receptor pairs between two different groups. So suppose we have two groups such as health and disease, denoted by H and D. We can just mimic the way that we constructed the joint similarity matrix W in the single sample case to construct what we call a couple joint similarity matrix V here with a joint similarity matrix from two different conditions, H and D sitting in the diagonal. And we put some constraint to enforce the correspondence in the off-diagonal block. So here the I is an identity matrix, and this beta is a scalar, is a tuning hyperparameter which enforces the correspondence between WH and WD. So this figure actually explicitly shows the construction of this couple joint similarity matrix. And here I would like to show you some preliminary results of my study. So we collect a data set, a public available data set from a GEO database, which the authors focus on inflammatory skin disease. In diagram A, we see that a bunch of ligand and receptor pairs have been validated by their experimental means. And in diagram B and C, we see that most of the ligand and receptor pairs validated by their experiments have been recovered by our pipeline, scTenifoldXct. They are inferred as a significant ligand and receptor pairs in our experiments. And we also visualize the up and downstream transcription factors associated with those inferred ligand and receptors. And we hope it will give biologists some new insights of what is actually happening between the different cell groups. And of course, in this data set, we also compare the performance of our pipeline scTenifoldXct with some state-of-art benchmark methods, including well commonly used cell chat and also single cell signal R, iTalk, and Connectome. It turns out in this data set, either AURC or AUPR, we achieved in both matrix analysis, our pipeline achieved the best performance. So that's basically the first my first project on cell-to-cell communication. And if you have any question, you can maybe ask me at the end of my presentation. So let's dive into my second project. So in this project, we were concentrating on the gene knockout. So it turns out the motivation is that the gene perturbation experiments are actually a proven powerful approach to show the role of genes. But usually the conventional knockout experiments usually require significant amounts of experimental and animal resources. which are labor intensive and time consuming. And the perturbation, which was developed in recent years, combine the CRISPR perturbation and the single-cell RNA-seq to perform the genetic screens. But we know that the creation of a large-scale CRISPR libraries still a major technical challenge, even for experienced experts working in the wet labs. So we ask whether we can develop a tool in an unsupervised way. So here we still want to use our single-cell RNA-seq data. So we always started with single-cell RNA-seq data. So this is the data from wild-type samples, and we will use computational tools, or if you have the ground truth of a gene record network, we can put the gene record network along with our gene expression matrix here as the input. And the thing is that we want to infer the wild type, and we want to infer the difference of the genes between wild type samples and knockout samples. The most intuitive way is that once we have the graph level information and the original design matrix, we can use the graph neural network to extract the representations of genes, actually the nodes in the graph, to do the following analysis. So here we chose to use the variational graph autoencoder to reconstruct the adjacent matrix from this inferred gene record network. And we will end up with the parameters of a pi distribution. Here is a simple bivariate normal distribution. So this parameter mu and the sigma are actually the parameters for genes in the graph. And the key idea is that how to construct the virtual knockout data, right? So here we propose to construct the virtual knockout data by simply removing the node, the target genes in the graph. So here, suppose this is the knockout genes, and we just remove the node and its corresponding links in our inferred gene record network. And then we put them into the pre-trained variational graph autoencoder again, and we will end up with the parameters for virtual knockout genes accordingly. And once we obtain the pair of wild type and knockout gene representations, the next question is that how we learn the similarity, or how we compare the differences between them. And since they are actually, we enforce them to follow a bivariate normal distribution, then we will simply can use the KL divergence, which is a matrix. It's probably the most straightforward way to compare the difference between two distributions to indicate the difference between these two settings. And from the distribution for KL divergence, we develop our non-parametric test. Actually, it's a permutation test to infer the significant level of perturbation for each gene. We call the top 5% highly perturbed genes as knockout responsive genes, and there are gene set, of course, and then we can input them to the prior knowledge-based gene function analysis to interpret our findings in a more biology language. So more formally, the VGAE, the variational graph autoencoder, is actually is actually a two-layer graph convolution neural network with inner product decoder. We actually didn't put anything new here, so I will briefly go through it. So this normalize adjacent. So suppose we have adjacent matrix A, and we will have our normalized tilde adjacent A, where we normalize the original adjacent A by its degree matrix. And we use ReLU in our encoder to put some non-linearity into our neural network. And we will infer the pi distribution in the latent space C. We assume a bivariate and normal distribution. So we will infer the mean and the standard deviation. Actually, the square standard deviation in the log space from the GCN. And in the decoder, since we want to reconstruct the adjacent matrix, so we just use the inner product decoder as our decoder. And the loss function of this VGAE is actually elbow. So people call it elbow, which is known for the evidence lower bound. So we want to maximize this lower bound. It consists of two terms. The first term is actually a reconstruction loss. It's actually minus mean square loss between the reconstructed adjacent matrix and the original input adjacent matrix. And the second term is actually a regularization term. It's the KL divergence between the conditional probability Z given the input X and A and our pi distribution for setting for this Z, which is a bivariant Gaussian distribution. And this beta is a scalar, is a tuning hyperparameter to scale the scale between these two terms. So first of all, we did some simulation studies. So there is a package called Sergio, which is a simulator for people who input the gene replay network, the graph as an input, and it will simulate using the dynamic model to simulate the gene expression matrix. So once we have this gene expression matrix, we can use our models and along with a couple of benchmarks to actually virtual knock out the target genes and evaluate whether we can infer the knockout responsive genes given the ground truth gene replay network. And it turns out that since our pipeline is running in a unsupervised way, we didn't require the perturb-seq data with massive perturbation labels. So we just compare the benchmark, including scTenifoldXct, which is a tool developed by our lab, and we also compare to two baseline methods, like use the Pearson correlation or the random uniform distribution as the output to compare these two classifiers. And it turns out in the evaluation of AURC and AUPR, our pipeline, our method achieved the best performance. In a case study, we collect public available data where people in the experiment knock out the TRIM2 in the neuron cells. So we collect the wild-type microglia cells and the knockout microglia cells in our study, and we virtually knock out the TRIM2 TREM2 in the wild-type microglia cells, and we obtain a total of 20 knockout responsive genes as the output of our pipeline, and we put this 20 gene set into the enricher. We did the Go process enrichment test, and we end up with those top 20 Go process terms. And most of the terms actually were mentioned in their original papers. And we also show that actually by query the protein-protein interaction graph, we show that actually most of the inferred genes are highly connected with each other under the prior knowledge. What is interesting is that we also set the gene expression level of our target gene TRM2 as a responsive variable, and the other genes as the explanatory variable in a multi-variable linear regression model. And we see that the R squared, actually the adjusted R squared is significantly higher than the model using random genes as the explanatory variables. So it proves that our inference actually those genes are expressionally connected with each other. That's pretty much about my research, and if you have any questions, just let me know.
永健，我刚才错过了呀，是结束了吗？还是怎么回事？我好像
我说完了。
OK，结束了对吧？我是网掉了吗？
是吗？没有吧
？OK，
没有是吧？没有，结束了是吧？
结束了，对。但是呢，在那个 chat 里边呢，在 chat 里边有同学问了几个问题。
对，我看一下。
我刚才就错了一小下，然后就回来发现等等。我们应该是没有没有外国人了，所以我们就直接用中文就没问题，应该。正好也是那个Q&A 环节。看看那个确实有2个问题，我看。第一个问题就是说，你那个 regulatory network 是怎么定义的？是好像就是 co-expression network 对吧？
对，这是一个好问题。其实我们是用的我们 lab 我之前师兄做的一个 work 叫 scTenifoldXct， 它是用 PC regression。OK。然后如果 PC regression 呢，我们是把每一个基因每一个 simple 基因一个单个的基因作为 Y， 然后其他的基因作为 X， 然后投到这个 PCA 的这个 low dimension space 上，然后去得到它的这个 coefficient。所以这个 coefficient 呢，在这里应该如果严格来说是不能够去 infer 这个 causality 的。不提供这个因果关系的，所以如果严格来说，可以把它理解为一个 co-expression matrix。
那这样子的话，它的这应该是一核你在线吗？不是，你再 comments 一下，看他是不是回答了你这个意思。 然后呢，应该是没有direction 对吧？它是 undirected。
是这个里面，这其实是个好问题。它是在这个 review 里面也有 review 说过这个问题，然后最后我们把original manuscript 里面的这个基因 regulatory network 改成了基因基因 for expression network， 好像是。
对，否则他确实会很纠结，
会
他不太喜欢让你用
不要误导
regulatory。
这个基因，可能要严格的去 infer 这个 causality， 还需要一些epigenetic 的这种 level 的 data。
对，义和说的对，说 PC 是 summary of multiple genes。义和你方便的话，你可以开麦，然后呢交流一下。
我的一个问题哈，就是说你讲receptor 跟ligand 啊，跟interaction 对吧？这有很多方法可以用不同的数据。你有没有比如说你或者你比较过，或者你从别人的文献里看到，比如说在原子水平 level 的，就是一边你搞 drug development 的对吧？他们搞了这个 ligand 跟 receptor 的interaction呢，有人发展的就是从有3米结构来看他们这是不是吻合 match。你这里吧是 RNA， 这是经表达的 data， 来看他们有没有那个当然也可以用蛋白质表达的 data。那你说准确度来讲啊，我们从准确度来讲，难易度也是另外一件事，对吧？一个是准确的度是这个问题，第二个吧，你在serotype里面对不对？不同的serotype那个3维结构，它是不是基本上相同，还是不同的serotype结构相差比较大？这两个问题。
对，这个其实是现在的一个趋势。它是首先我承认是我们用基因 expression 的 data 其实是实际上是把它作为一个 proxy， 是作为一个代表，是作为一个indirect 的这种代表去实际上去推测实际上这个 protein 的 expression。其实 ligand-receptor 理论上是还是应该去测这个实际上它每个 cell 之间的这个 protein 的 expression level， 所以单纯的从 single cell RNA-seq 上面来说的话，我们只能去推测一个大概可能的这个 ligand-receptor pairs， 里面可能会包括一些 false positive 的信息，这个可能要配合后面的这些 proteomics 的技术成熟，然后我们能够更能够去排除一些这个从这个 data 里推出来的 false positive 的 ligand-receptor pairs。然后刚才您说的这个从protein 的这个 structure 的角度去推测这个可能发生的 ligand-receptor pairs， 我觉得他们关注的点可能跟这个有点不一样，他们可能更希望知道的是在一个 3D 的 structure 上，一个什么样的 ligand 和一个怎么样的 receptor， 他们更能够结合到一起去做这个some communication 之后可能导致的这些 pathway 发生的这些事情，所以他们研究的点可能是怎么样去从根本上去 design 这个 protein， 可能他们这个关注的点和这个在这个基因 expression 来我关注的点可能稍微有一些不一样。
对，那你这个不需要那么精确，得要大概的看看它就肯定
是这个对，这个只能做到这个程度。如果要非常精确的话，我们还是得用那个 fluorescent 的，然后去测一个特定的 ligand-receptor pair，通过实验的方法去验证。
OK， 但是那个永健，是不是你在你这个分析中，你这个 ligand 实际上也是细胞膜表面的一些蛋白，你是 limit 到这些基因上，对不对？因为你是从数据库里来的，我可以这样理解吗？
对，我是从数据库里来的，但是我们这里收集了很多个数据库，最后整合了一个 database， 所以应该还是目前在市场上算 relatively large 的一个 database。
OK， 那么，熊老师，就是如果说我们用那个3D structure 去做 simulation 去看它这个 ligands 和这个 receptor 之间的那个 binding 的，熊老师，这个事情也可以做，对不对？有没有？据你所知，有没有方法，熊老师？
方法是有噻，对，现在就是麻烦噻。
是计算量大？
计算量，对吧？计算量肯定大些，结构对吧？研制水平上的啊，这当然你也可以不研制水平，也可以结构也可以做，因为现在这个取决于你的这个计算的量啊，计算的难易程度，对吧？它跟准确度是息息相关的，跟提供信息相关的。你计算的量大，提供的信息就多，准确度就比较大。你比较马虎，用点数据做一下，那它提供的信息误差就比较大，这个东西相互相成的。
那个好，那个一和他又 follow 了一个问题，他说怎么是得到这个基因 coefficient？我猜应该是2个基因分别放在那个我们那个 formula 两端，然后再加上 PC，是这个意思吧？
对，
还是
我回答一下这个问题，就是假如你要 P 一个 gene 的话，我们是把其中的一个 gene 设为 target variable，然后 n_p 减 1是这么多个 gene 作为 x，然后相当于做一个 linear regression， 但是因为我们是在这个 lower dimension embedding 上做的，所以我们在乘以它的这个 PC， 就相当于乘一个 transpose 的那个PC component。去把它 map back 到那个 original 的 space 上，所以这样的话，我们就会有一个原始的 P 维的coefficient 作为这个2个基因之间的这个 interaction。
然后他最后的一个问题就是说，这是一个undirected Gaussian graph 还是一个 directed Bayesian graph？ 才应该是前端吧，应该是。
这是一个好问题。其实，如果我们用 PC regression 来做的话，你想一下，如果我们每次把其中的一个 individual gene 设成 Y 的话，我们其实最后会得到一个 direction 的 graph。然后，如果大家熟悉这个 manifold alignment 的话，其实是我们传统的是用 eigen decomposition 去解这个 joint similarity matrix 的 Laplace matrix。如果要去做这个事情的话，那其实它数学上它是需要一个对称的 matrix， 这个 graph 需要是对称，所以在最初的这个 scTenifoldXct 这个 work 里面，我们是强行把它作为一个对称的。这个对称就是从数学上其实就是很 simple 的，就是 transpose 加上这个 original 除以2了。但是在这个 work 里面，我们用 neural network 去解这个，我们其实并不需要这个 symmetrical 的这个 constraint， 所以其实理论上来说，我们并不需要这个这2个 network 是对称的。
我补充一句哈，就是后面有人写的文章啊，就是说在这个用拉普拉斯方程来出这个 manifold 这个 alignment 它呢对有向网络，它最近这2年发展新的方法，不需要这个矩阵是对称的 symmetric 的，对吧？但你这里呢，因为我没有仔细去想，你比如说你这里就不需要对称，对吧？你是来看看，就是说这种方面讲怎么样？反正你也是一整嘛提出来，对吧？那别人也有人提出，就是说不需要它的 matrix 是 symmetric， 那它当然有一些其中有一些modification， 改动，就是说，如果是有机会的话，金浩因为你做这个事情吧，可能我觉得manifold的 alignment 对吧，是要解决有向网络的它的这个 alignment 对吧？就是这1年以前吧，因为我在学习这个问题的时候我看到查到是文献，他们呢对非对称性的有向网络，他们也有些解决办法。
我来请教2个问题，然后那大家再有问题的话，就是直接开麦哈。我2个简单的问题，那个永健，就是第一个就是说，我还是没有完全 get 到你的这个第一个方法的所有的 assumption， 就是说你能不能再给我们快速说一遍，你这个能列举一下所有的 assumption 是什么？
行，我快速过一遍我的 pipeline， 其实就是我用 PC regression 得到2个 gene record network， 或者说 gene co-expression network。然后我们希望用 manifold alignment 去 align 这2个 network 到一个unified space 上，然后在这个 space 上，ligand-receptor 比较 靠近的，他们的 distance 比较近的是我们认为是significant ligand-receptor pairs， 他们正在 talk。但为了完成这个任务，我们需要 design two neural network， 然后这2个 neural network 我们需要去学习的是这个 joint similarity matrix 里面包含的 similarity 的信息。loss function 就是说我们需要去 maintain 这2个gene replication network 里面的每个 node 之间的 similarity 和我们做出来的这个 correspondence， 它们之间的这个每个基因从 cell type A 和 cell type B 里面 construct 这个 correspondence 的这个 inter similarity， 我们需要去 maintain 这三个 similarity， 然后把它们project 把它们投影到这个 lower dimension 上。后续的就是我们去学习这个投影之后的每个点的 similarity， 然后就会有 distribution， 然后 distribution 上面去 infer 这个 significance。
明白了。那么在这个地方，就是说，我还是比较因为我们在做这个时候，老师会担心，就是我如果说有 ligand 和 receptor 这种缺失，因为我老是怀疑那个数据库它不全，有大量的这种 interaction 并不在那个库里边，这种大量的 missing， 你觉得会影响这个结果吗？
我从我自己的观念来说，我不代表任何人观点，我觉得肯定会的，因为现在这个 ligand receptor 库大概我知道的大概是1000到2000个 pair。
对，
但是实际上之所以有那么多的 biologists 在研究这个东西，肯定是这些不同的 cell 之间，这个 ligand receptor 具体是什么，肯定是有差别的。所以在这个领域也有很多人提出说，你要做这个 cell communication 的话，你需要去做一个 cell specific 的这个 pipeline， 而不是一个 generalized 的 pipeline。
我也觉得这样。
任何的2个 cell type。对，这个是这个，但目前我们并没有市面上我并没有看到一个这个 cell specific 的 ligand receptor database， 所以目前我们就是按照这个 generalized database 去做了。
OK。我觉得这个东西可以继续 follow， 因为最近有个技术叫 surface norm， 就表面组学，他们实际上可以解决这个问题，把所有一种类型细胞表面的ligands 和 receptor 全搞出来，这样子的话可以非常好的 support 你这个工作。第二个，我想请教一个简单的问题，就是说我看到你第2个是敲了这个 trim2， 它是你这个库是只敲这一个基因对吧？只敲一个trim2， 还是说它可以敲好多基因？
它理论上来说，其实只要我们得到了一个 ground truth 的 gene replication network， 或者说我们用任意的 computation 的这个方法得到了这个 gene replication network 之后，我们其实可以按照我们目前的这个比较 naive 的比较 straightforward 的way去敲这个基因的话，我们其实可以敲掉任意一个基因。
如果同时敲2个和多个，你这个算法还依然适用吗？
在我们的文章里，我有一个 case study， 其实是验证了这个 double knockout。double
knockout， 明白。
对，是敲2个基因。然后这个事情啊，就是当然现在没有，我目前没有发现有什么人去做这个事情，就是这个 virtual knockout 的 inference， 我们现在我们在这个 work 里面想做的是提供一个纯粹的 unsupervised way to infer， 所以我们只需要 wild type data， 但是像不知道如果您比较关注这个 single cell 的话，可能知道现在有一些Fabian Size 在德国的那个组做 single cell 的那个组，他们组做了一些 work，是推断这个 perturbation effect， 但是他们是 train 这些 model 在一个 unsupervised way， 所以他们需要大量的 data， 他们的 input data 可能会是 perturb-seq。我们需要很多的 gene expression pattern with 这个 perturbation labels。然后这样去推断它这个事情，所以在我们的文章里，我们并没有直接的跟他们的pipeline 去做一个直接的比较，因为我们这个地方并不需要 perturb-seq 这个 labels。然后后续的一些work 的话，当然就是说这个我们现在是 straightforward 的去直接去 remove 这个 node， 可能并不是一个比较好的方式去去 simulate 这个 knockout。比较好的方式，我觉得可能一个比较好的方式是我们可以 model 这个 graph model 这个 distribution。这个 graph 的 distribution， 比如说一个高深，比如说一个 graph 的老寿，或者说那个高深 graph model 之类的东西。所以一旦我们去 knockout remove 一个 sample 在这个 multivariant 的高深 distribution 上面，其他的 node 也会跟着联动。这样可能会是一个更 probability 的 way 去模拟这个knockout。
明白。那个 TREM2 的 knockout 是在搞 Alzheimer 吗？是在那个课题是 for Alzheimer 吗？还是 for 什么？还是纯粹的一个？
这个 original paper 我现在有点不记得，我记得好像是对您说有可能对，他们研究的是阿兹海默。
那么他有没有比较一下我 knockout 之后 knockout 之后有没有和 Alzheimer 的那个病理的他那个情况的比较？有做吗？
这个我现在不记得他们
没关系，
可能我也得去查一下。不过我们这个 term 里面应该有一些关于阿兹海默和他那个 lipid generation 的事情。
OK， 好的，其他的听众有没有什么问题？我们可以再提。
我来做一个一般的 comment 啊，对所有参加这个大家会议的啊。那现在这样的，现在我们数据已经有很多，对吧？能法搞个理念模型其实是可以做的了，就是说